All right, hello guys. All right, let's get back into it. I'm going to start recording the neuroanatomy portion. So the chapter 3 slides, they're like 64 slides. I'm going to start on the central nervous system when we start talking about neuroanatomy that we've done so far. We'll talk about cranial nerves, the peripheral nervous system, and spinal cord, and maybe brain imaging. It depends on how long the recording goes. So let's go ahead and get started. A lot of this we've already talked about, but again, the idea is to have all of neuroanatomy concisely into one lecture that you can go back and review. So rather than just doing new material, I wanted to do all of the neuroanatomy in one recording. Okay, so let me get this going. All right, so as I mentioned before, the subdivisions you don't need to know. Telencephalon, diencephalon, all of that. I do want you to know the major forebrain structures, and we did talk about ventricles. So the lateral ventricle is on the side of the brain. They flow, the cerebral spinal fluid flows into the third ventricle from the third via the cerebral aqueduct, which you can actually see here on the sides. If I get my mouse to be big, you can see right here through midbrain, and then kind of behind, through and sort of behind, well, we actually haven't talked about midbrain yet, but it's going to be going through midbrain and hindbrain. And so the fourth ventricle is on the very bottom here, where it's basically between brainstem and cerebellum. So orienting yourselves by the forebrain, midbrain, hindbrain structures based on where the ventricles are. These are not all of the structures, right? So I uploaded a document for you guys to be able to see all of those structures. These are a lot of them. But again, look in the comments because a few I added that are not on this list. The periaqueductal gray, the colliculi, the basal ganglia, and the two glands that we'll talk about. So don't worry about this minute. If you want to know all the structures, there's a separate document called Brain Structures List, I believe. That's up on Canvas. But in general, the way that we're going to split the brain are all of these forebrain structures. So forebrain in blue, that's the big hunk of the brain that in humans and primates and mammals in general is very large compared to the other mid and hindbrain structures. You can see just how small midbrain is. We're only going to have 3 structures in here, and then hindbrain really constitutes just another 3 structures: cerebellum, pons, and medulla. So the majority of the structures that we go through are forebrain, so cortical structures that we've talked about thus far, and then we're going to talk about subcortical structures underneath the cortex, hence subcortical underneath the cortex. Okay. So again, we've talked about this, but I'm just reiterating it. Gray matter, when we talk about, I'm losing gray matter, or just in general, it's a very popular term that we refer to when we talk about gray matter, meaning brain matter. Much more specifically, that means the cell bodies of the neurons as well as glial cells. And that's what really constitutes the cortex, right? When you look on the outside of the brain and you see these bumps and grooves, meaning the gyrus, the gyri, and the sulci, right? Plural sulci. It's showing here as a darker color because that's where the majority of the neuron cell bodies and the glial cell bodies are, right? Unless we're talking about an astrocyte. Astrocytes are going to be in the cortex too. But for example, oligodendrocytes, they're going to be where the axons are, and the axons are underneath the cell bodies. So this is a nicer view to kind of really illustrate. On the outside is our cortex. Underneath is what we then call the white matter, which are the myelinated axons. So it does appear white because fatty tissue is wrapped around those axons. And so basically we have neurons with their cell bodies in the cortex and then sending their axons, for example, over to the other hemisphere or down to spinal cord or from frontal lobe down to temporal lobe, however the direction wants to go, right? In class today, we talked about the arcuate fasciculus. Again, I'm not expecting you to know that name, but it's this connection between Wernicke's and Broca's area, right? When you're understanding something, Wernicke's area for speech comprehension, and then Broca's area, there are axon fibers that send Wernicke signals to Broca's area. So those would be white matter, right? This is, for example, a a superior temporal gyrus that communicates with an kind of a more anterior in the frontal lobe structure. So basically just think of white matter as various areas that are sending their connections wherever they need to go. Okay. What else do I have on here? Yeah, two hemispheres covered by the cerebral cortex. And then cerebral, we often call this the neocortex, and that's just on the outside. Neo, because of evolutionary reasons, it's the newest cortex that expanded and evolved, and where really our higher cognitive functioning is that not all animals share with us being humans. Okay. And so this was just an illustration of that idea of the cortex on the outside, like the bark of the brain, versus inside, where we get a lot of the big subcortical structures. Those are really big clusters of neurons. Like the amygdala is a cluster of neurons. And then they have axons that go and send out information. Or, for example, somewhere in the sensory cortex, it brings information to the amygdala. But the amygdala itself, these different subcortical structures are big clusters. It's not cortex, but it's other clusters of neurons that are doing various forms of communication, or rather, they're all doing neural communication, but important for different behavioral functions. And we started talking about that a little bit, or we will start talking about that next class. But I'm going to go over it here. Okay. All right, so the four lobes are frontal lobe, right? We have the dividing line between frontal and parietal is going to be the central sulcus, right? So in purple here is going to be our frontal lobe. Behind or posterior to the central sulcus is going to be the parietal lobe. And then, as we talked about, we have these functional areas in each of the lobes that do very particular things. So we have, again, if you're looking at my cursor here, going like, I'm trying to make this guy bigger, come on. Well, I hope you can see. This gyrus, meaning this bump, right, the outside cortex, running from dorsal down to ventral, this is going to be the primary motor cortex in front of or anterior to the central sulcus. The one behind it, this gyrus, and again, each person's anatomical configuration looks a little bit different, but overall, we're kind of going to have this map of the brain where behind the central sulcus we have the primary somatosensory, right? And you can see that up here, primary motor, primary somatosensory. So somatosensory being touch, right? The sensations you get in your skin that tell you temperature, pressure, and pain. Okay, what we just started talking about is the occipital lobe. Occipital lobe in the back, or you can see over here, not very big. It's more if you turn the brain and look from the back side, that's the majority of the occipital lobe that you're going to see there. And in the very, very back side of occipital is primary visual. So again, when it comes to functions, occipital lobe is just vision. Just vision. It takes a lot of information processing to put vision together. And we're going to talk about that when we do the vision chapter. Parietal lobe, space, right? Spatial awareness. If we go more abstract, your awareness of yourself, meaning kind of self-awareness, consciousness, the fact that I have my own consciousness and somebody has their, a different kind of consciousness than I do. But at the same time, we can kind of recognize each other, that we are both aware. Things can get pretty abstract pretty soon, or pretty quick. But nevertheless, space, spatial, configuration, awareness, as well as movement. So when you see something moving through space, or you can imagine yourself walking through your apartment or your home, right? Because you have an internal map of what that space looks like. Okay, and then frontal lobe, right? It's a lot of things, but think your main cognitive control. Control over emotions, control over actions, control over decision making, and future planning, right? Which then end up being reflective of your personality, how you would act in a given situation. Okay, and then the last lobe is going to be temporal lobe in green here. Temporal lobe, and inside, or as part of the temporal lobe, We have primary auditory cortex here. So hearing. And then don't worry about this yet. We'll talk about the fusiform face area. This is actually one of those areas that helps decode faces, which is really cool. But you can't see it here, but roughly here, right behind, meaning posterior to the auditory cortex, we'd find Wernicke's, the other language area. And I have a separate slide on the language areas. Okay. Nothing new information here, just showing you different views of the lobes and the cortices. Oh yeah, and the lateral fissure, I did forget to mention that. Although you probably saw that on the animation on the last slide, but lateral fissure runs along here. It separates frontal from temporal as well as temporal from parietal. So kind of that like running up along your ear. Okay, the corpus callosum, we did talk about this briefly, but this is one of those areas that I definitely want you to know. It's axon fibers. It's not an area necessarily. It's not a nucleus or a cluster of neurons. It's axon fibers that are crisscrossing and allowing the two hemispheres to talk to one another. So even though you have a left brain and you have a right brain, and they both are, in most cases, doing similar functions. Again, remember, sorry, I'm a little congested. I feel like I keep having to breathe in. What was I saying? Pretty much every function except for language is bilateral, right? Both hemispheres serve similar functions, motor, sensory, whatever it is. But remember what I had said, that all motor and sensory functions are contralateral to the brain area. So any motor function for my left brain is controlling the right side of my body, and excuse me, the right hemisphere is controlling all of the left motor action in the left side of my body. Same with sensory information, whether it's coming in through the eyes, ears, taste, smell. Actually, not smell, that is the one exception. All sensory information, including touch, goes contralateral to where it came in. Right, and now you're going to be like, well, we have two eyeballs and we have two ears, right? How's that work? We'll get there. We'll talk about vision. We're not going to talk about hearing. But one exception to that rule, where the sensory information stays ipsilateral, is smell. Right, so if you plugged one nostril and you just breathed in through one, it would stay on that side of the brain, ipsilateral. And vice versa, if I plug this nostril and I let it come in through here, it's going to enter my left hemisphere. All other sensory processing goes contralateral. Okay. Yeah, and when you look at a view of the corpus callosum, right, in this picture here, you can see that post-mortem image, kind of really seeing where those fibers are going. Here you can see that really that corpus, that bridge, corpus meaning body, this very large body running anterior to posterior, right kind of on top of the brainstem. The brainstem being, the majority of this chunky bit here. You can see kind of where all of those crisscrossings of the fibers are going. You can see it again here. Okay. All right. So we started talking about our sensory regions, and taste and smell, you don't need to know where these are, because they're going to be in frontal lobe and they're going to be embedded, and it's kind of hard to find them. So the three, I want you to know all of these regions in terms of what they're called, right? It would be the olfactory cortex or the gustatory cortex. But I do want you to know these top three. So we talked about touch, somatosensation, right? Our primary somatosensory cortex. Our primary visual cortex and our primary auditory cortex. One in each lobe, right? Parietal lobe has one, occipital lobe has one, and temporal lobe has one. And motor is in the frontal lobe. So generally we think of frontal lobe being any kind of action-oriented processing, right? If you're trying to control how you're acting or control how you're feeling, planning, thinking about acting. The frontal lobe in general is considered, or almost the front half of the brain is usually considered action-oriented, whereas the back half of the brain is often thought of as sensory processing, right? So sensory motor, all of these areas are talking to one another to decide, how am I going to act in this environment? Okay, and I mentioned that all sensory information is contralateral. And this is a thing not to forget. So even though we have primary sensory and primary motor regions, everything else, everything in between, is considered an association cortex. And again, as we go through the class, we'll talk about those more specifically. All right, so we talked about the body map in the brain, right? So when it comes to motor cortex, you can see motor cortex runs the length of the left brain, the length of the right brain, right? Again, left brain controls right body, right brain controls left body. And same with somatosensory, it's running in both hemispheres, right? So even though it's showing here as if this was left brain and right brain, just be aware that it's in both hemispheres. The homunculus, right? So this guy is the brain's representation of your body. Giant hands and giant face and lips and tongue, because that's where we spend the majority of our cortical resources. And so this idea of somatotopic organization is the idea that we have a point-to-point correlation. A point on your body corresponds to a point on your brain map, right? So somatotopic, meaning the topographical organization, so to speak, right? Okay, language. It's one of the only areas really that we're going to talk about that is only on one side of the hemisphere, or in one hemisphere only. So Broca's area for language production, meaning speaking fluently, as well as Wernicke's area, being able to understand when someone talks to you, in two different lobes, but they communicate with each other a lot. So we went through and we, tried to draw out where those guys are, Broca's in frontals, and Wernicke's in temporal lobe. Okay, so I want to walk through this. This is an example of what would happen when you disable the two hemispheres from being able to talk to one another. So these are called split brain patients. And I explained this in class that it used to be when we had epilepsy patients where it was so bad that wherever the seizure activity starts, it kind of starts in one hemisphere and spreads over to the other hemisphere. And it spreads via the corpus callosum, those like nerve bundles that are crisscrossing, right? So the idea, and you can actually see this here in this really beautiful image, you get this from MRI. When you just calibrate the MRI to just look at axon fibers, you can really see this right here is the corpus callosum. This is a coronal slice, right? And out here would be cortex, because that's where the nerve fibers end. But you can see in some nerve fibers are going down. The color coding usually indicates the direction that it's going, but you can see left to right and right to left. Huge amount of fibers. So what we do is we sever, meaning we cut those fibers so that when the seizure is starting in one hemisphere, it's not going to go jump over to the other hemisphere. So we can do really cool things that we can study with split brain patients, where we really see this, that awareness is almost stuck in a hemisphere. So let me just walk through this really quick. So, and part of the interesting idea of awareness and being able to be able to speak about your awareness is because of language. So let's just say this participant, this is a split brain patient. They have their left nostril plugged and a perfume is presented in the right nostril. So if you remember when I mentioned smell is something that is ipsilateral. So if the left nostril is plugged, that means the perfume comes into the right nostril, the olfactory nerve, and it stays in the right hemisphere. So perfume with aroma of rose is presented to the right. Now, because it can't move over, the corpus callosum was severed, so right hemisphere can't go and tell left brain what happened. And in the left brain is where we have our language centers, so the person would end up denying that they smelled anything at all. They weren't aware of the fact that they smelled anything because the left language brain couldn't receive any information from the right brain that actually smelled the rose. So if they were to try and communicate verbally, they would say, I didn't smell anything. But what you can do then, instead of asking the person to say what happened, you can let them use their hands. So again, right brain controls the left side of the body. So instead you tell the person, point to the object that you just smelled. And then they will be able to, with their left hand, which is controlled by the right brain, with their left hand they can go and select the rose and indicate it. So the information is stuck in the right hemisphere. So anything having to do with the right hemisphere, such as controlling your left hand, any left body, you could act on it. But then if the researcher asks, why did you pick up the rose? Language center never got that information. It would start BSing. You'd be like, oh, I don't know, it looked pretty. They just would not be able to say it's because that's what I smelled. So that's just one example, but this really interesting split of consciousness, and not just split of awareness, but almost split of consciousness. So if we have time in class, I'm going to play this for you guys. It's just a 2-minute video of Ramachandran, world famous why am I blanking right now? Neurologist, not neuroscientist, neurologist who studies different case studies of neurological damage that people have. So hopefully I can play this in class because it's really cool, but I did put the link in here if you want to watch it yourself. Okay. So those were all of our cortical structures. I'm sorry, I'm congested, so I feel like I'm having a hard time breathing while I'm talking. This is why I couldn't record yet. I'm doing better, but I'm still struggling through lecturing. Okay, so we talked about our 4 lobes. We talked about functional cortical areas in those, and now we're going to start looking underneath the cortex, right? So inside of the brain, we have a lot of weird, loopy, alien-looking structures, so we're going to start talking about those. The first one to talk about over here is the thalamus. So the thalamus in blue, you can see it's inside, it's underneath this structure here that we'll get to. But the thalamus, if I were to rotate the brain and look at it coronally, like a coronal slice, you have 2 of everything. I'm going to say the hippocampus, the amygdala, the thalamus, but remember there's 2 because everything's bilateral. So I don't just have 1 thalamus, I have 2 thalami. We still just say the thalamus, but we have 2 of them. And they actually sit right next to each other. There's a tiny, tiny little bridge that connects the 2. But so very medial to the brain, right? It's kind of like smack in the middle of the brain. And I'll show this more explicitly on another slide. But thalamus, and then down here into these hindbrain regions, this is what we call the brain stem. So thalamus being kind of the top of that brain stem. Okay. Function of the thalamus. The main sensory relay station of the brain, right? So we talked about our senses, hearing and seeing and touch and all of this. All of that information, when it comes in from the cranial nerves, right? Your optic nerve, your auditory nerve, peripheral nerves in your hand, all of that information, before it goes to primary sensory cortical areas, it has to go through thalamus. So think with your eyeball. Well, you see with your eyeball, but think for a second with your eyeball. You get visual information into your eyeball, which is a peripheral nerve, a peripheral cranial nerve. That information doesn't go straight to primary visual cortex. It first goes, it first goes through the thalamus, and then from the thalamus all the way to the back of the head, which is where primary visual cortex is. Same with hearing. It's going to go kind of into a deep brainstem area and then through thalamus and then out to primary auditory cortex. So even though auditory cortex is kind of right next to the ear, it first has to go into thalamus and then back out again. Same for all of your senses, again, with the exception of smell. Smell is the only one that doesn't need to go through thalamus. Smell, there's a lot of exceptions to smell. In part because it's evolutionarily one of our oldest senses, and for most animals, sense of smell is going to be the most informative in terms of threat detection or mating, right? Pheromones. Humans, of course, we can detect pheromones as well, and we respond to them, and we don't like icky smells, but pretty much every other animal in the animal kingdom, that's way more valuable information for them. So when we talk about the amygdala, which we actually will in this slide here, and you've probably heard of me talking about it before, but the amygdala is really kind of the main emotional information signaling. If any kind of emotion is detected, this area of the brain, the amygdala, is going to light up and connect to a whole bunch of other areas saying something emotional just happened, right? What are we going to do? Are we going to look at it more? Are we going to run away from it? What are we going to do? So when we smell something, let's just say we're not a human right now, we're a rat or we're some other animal. Well, let's actually go with the example of a rat. If a rat smells cat urine, that is an extremely aversive smell for them and will immediately put them in a fight or flight state, right? So smell doesn't go to thalamus. It actually has a direct input into amygdala, so that it can very quickly tell the rest of the brain, I detected something I need to get away from. Or a rat is walking around doing its thing and it smells pheromones, right? Then it's like, ooh, I can't miss this opportunity for a mating partner, right? So smell from the olfactory bulb will work its way to the olfactory cortex, which again, we didn't talk about where that is. But it also has direct connections to amygdala, which no other sense has. All of the other senses, they have to go through thalamus, then out to the primary sensory cortical area, and then it goes to amygdala if it has connections then to the amygdala. So that's why it says here, sight, sound, touch, and taste. It's leaving smell out because it has direct access. It doesn't need to go through thalamus. It has direct access to these other areas that it needs to go to first. Okay, what else was I going to say? Oh yeah, so with smell having that direct access to amygdala and activating emotional memories, this is why you so easily can have this experience that say you haven't smelled something in years and you like go home or you smell like a book or you smell a fruit, I don't know, something that reawakens an old memory of yours, right? And it's like it just smacks you in the face. You smell it and you're immediately transported. You have really this visceral response where that memory, it feels like you're reliving that memory again. And that is because of that very strong connection of smell to amygdala that goes then and activates your memory circuits. It brings that back, right? So that you're fully aware of what this smell means. Okay. It's cool stuff. All right. Thalamus sensory relay station. Oh my God. I wish I wasn't so congested. I should have taken like Flonase or something. Okay. There's an area underneath the thalamus that is aptly called hypothalamus, hypo being under, right? So it literally means under the thalamus. So over here, hypothalamus, main function is body regulation, right? So think blood pressure, body temperature, to some degree wakefulness. We're going to have other areas that are more important for whether you're asleep or awake. Thirst, hunger, basically, it's your main communication point between brain and body, right? If the brain needs to know something that's going on with the body, especially your organs and that kind of stuff, or making sure that your hormones are in order, say say you did smell pheromones, we can go back to being a human. Say you smelled pheromones and all of a sudden you're like, ooh, important information, right? Maybe your hormone levels change. Maybe you're preparing to like go talk to that person, be like, ooh, I'm attracted to them, whatever it is. The hypothalamus, and then maybe you start blushing, your blood pressure goes up, your heart starts speeding up. All of that is because the hypothalamus is regulating your body to reflect what your brain is processing. Right? So think, basically keeping homeostasis of the body. And because of that, because it regulates, I don't have it on the slide here, I'll have it on another one. It's the main regulator for our hormones. So hormones, blood pressure, all of this stuff. It then also has very important implications on what we call the 4 Fs. These are survival behaviors that if you're going to do one of these survival behaviors, you need to make sure that brain and body are linked up and you are doing the appropriate response. So the 4 Fs being fighting, fleeing, feeding, and mating. Right, so basically there's a threat. I need to either fight it or get away from it. Those are 2 Fs already, fight or flight, fighting or fleeing. And then the other 2 are more for, let's just call it low-key survival, right? So if you don't eat, you die. If you don't procreate, then eventually your species dies out. So it's more of a low-key survival, but all of them are still survival-oriented. What else to say about this? Well, I'll have another slide, so I'll talk about it more. But think your endocrine system, hormones, and glands, right? You have actually 2 glands in your brain. I'll talk about that. But you also have glands all throughout your body. The hypothalamus controls all of the glands. Okay. So that was the thalamus and the hypothalamus. Now we have 2 more over here. So, hippocampus. We'll come back to hippocampus. But let's talk about amygdala real quick. Has a reputation of being the fear center. It certainly does fear processing, but it does a lot more than that. So I like to call it the emotional alarm system. So if your brain detects any emotional content, whether you see it, you hear it, you smell it, or you think about it, it can be internally generated. Like you're sitting there and all of a sudden you remembered you forgot to submit an assignment and you start panicking, right? Amygdala is going to be like, oh no, she forgot, right? So any kind of information that is salient, meaning important, the brain interprets it as valuable information, or just purely emotional, right? But you could argue emotion is based on how I interpret the situation, because some people love roller coasters, other people hate roller coasters, right? So roller coasters isn't necessarily inherently good or bad, it's how the person interprets the experience. So any emotional content, anything that is like new and shiny and weird and out there, right, where the brain doesn't really know what to do with it, amygdala comes online for all of that. And we'll talk about that more when we do emotion. But for now, think emotional and salience. Salience just meaning important, it stands out. The ooh, shiny effect. Okay. So the amygdala and several other structures compose what we call the limbic system. So I'm not going to have you go through all those areas, right? Unless you're taking behavioral neuroscience, which I teach. There's a much bigger list for this, but for this class, what I want you to know, the limbic system is the amygdala. We just talked about that. It involved in emotional processing, novelty, salience, and the other structure is the hippocampus. So the hippocampus you need in order to make new memories. If you look over here, it's kind of a frontal side view of the brain. This little red bulb here is the amygdala. Again, we have 2. So we have one over here, one over here. Both the hippocampus and the amygdala are in the temporal lobe. Right, so if you've been like kind of zoning out, Now is a really great time to wake up and write that down, because most of the subcortical structures you don't really allocate to a lobe. It's just kind of like in there. But these two are off on the side. They're off in the temporal lobe. So it's not showing it very good here, but this would be temporal lobe. Inside of the temporal lobe, on the front portion is the amygdala, does all of our emotional processing. And then right behind it is this wormy looking structure called the hippocampus. And it runs kind of half the length of the, well, not half, but a certain degree the length of the temporal lobe. If you are missing the temporal lobe, or if you are missing the hippocampus, you cannot make new memories. Say, patient, if you guys remember patient HM, we now know who he is because enough time has passed. He was having seizures in the area around his hippocampus, so he had his hippocampus removed, and then he couldn't make new memories. And again, in the memory and learning chapter, we'll talk about it a little bit more. He could remember everything that happened before the surgery. He can remember his childhood and everything in his adult life. But since the surgery, he wasn't able to make any new memories. So memories are not inside of the hippocampus. Otherwise, if he had it removed, he wouldn't be able to remember anything. But it's required to form new memories. Okay, so those are the only two subcortical structures of the limbic system. Think the limbic system, emotional memories. Right? Most valuable memories are emotional ones, except for maybe a lot of the stuff that you have to learn in school. It's just like pure memorization, right? But in our experiences in everyday life, the things we remember best are the emotional ones. Okay. Just checking my time. I think we're almost at 40 minutes now. Okay. Basal ganglia. I'll stick on this slide for a second. Well, it's actually on the next slide. Let's go back here. Sorry. Okay, in blue here, the basal ganglia are 3 different structures inside of it, but it's this entire blue thing here. This big round thing and this tail coming off of it. So the basal ganglia contain these 3. You don't need to know what each of these 3 do. Just know that the basal ganglia are a cluster of 3 different structures. The caudate nucleus, the putamen, and the globus pallidus. So these 3 constitute or make up the basal ganglia. They basically help control movement, right? Willful movement, initiating movement. Let's say you learn to ride a skateboard or snowboard for the first time and you've never done it before. You're gonna need your basal ganglia to do that. Otherwise, you're never gonna learn it. So when you're trying something new and you're learning it, there's a certain type of learning that is called, that ends up being stored as what's called procedural memory, right? So basically think kind of muscle memory, right? When you get so good at something that it's automatic and you don't have to think about that, it's thanks to the basal ganglia. So again, until we do learning and memory, don't worry about it too much right now, but know it's important for the control of movement and for initiating movement. Later, we're going to talk more about learning movements and things becoming habits. Okay. We've talked about Parkinson's before when we talked about the degradation of dopamine neurons. Dopamine neurons are very much needed in the basal ganglia. So if basal ganglia is not getting enough dopamine, think of it as fuel. If we don't have any fuel, then the basal ganglia are not going to run properly. So this is why in Parkinson's, it's not just because they don't have enough dopamine. We don't have enough dopamine in the basal ganglia. And this is where you get all of those motor deficits, right? The tremors, the shaking, the postural, hunching, difficulty initiating movements. So most of Parkinson's symptoms are going to be these motor behavioral symptoms. Okay. Thalamus, as I mentioned, the sensory relay station. You can see here, nice round bulby guy, right? So really quick, between basal ganglia and thalamus, where are these guys situated? If you remember, I said the thalamus, again, you have 2. They're right next to each other in the middle of the brain. The basal ganglia, again, we have 2. We have 2 caudate nuclei, 2 putamens, and 2 globus pallidi. but we have two of them. So if these are the thalami, then around that we have the basal ganglia. So the thalamus is medial to the basal ganglia, or the basal ganglia is lateral to the thalamus. And you can kind of see that here in this figure. The blue is the basal ganglia, and the purple is the thalamus in here. Okay. Yeah, it's another view where you can really see that relationship, right? In thalamus, these two pink guys in the middle, and around, kind of like a shell around it, you have the basal ganglia. Okay. Hypothalamus, we talked about hypothalamus just being a little bit more explicit here. So where the name comes from is literally underneath the thalamus. It controls our autonomic nervous system, right? So if you think about thirst and hunger, any kind of like organ thing, internal organ, as well as that fight or flight response, the parasympathetic nervous system response, rest and digest. I'll talk about that a little bit more. Probably not in this recording because it's already starting to get long. And then the endocrine system, meaning your hormones. Hypothalamus controls all of your hormones. And again, relating to the 4 Fs, feeding, fighting, fleeing, and mating. There's another word for mating that starts with F. If you have questions, you can ask me. But it's a crude way of saying mating. Okay, mating being, do I really need to explain that? I sometimes have people who don't know what that means. Just ask me if you don't know. Okay, here we can see the round ball of the thalamus, right? So again, over here, this little button here that's called massa intermedia. Again, you don't need to know those terms. I'm trying to be very explicit with which terms that you need to know. And often I know I have pictures on here where a whole bunch of labels. Don't worry about all the labels. Just worry about the terms that I tell you guys. The hypothalamus. But this is actually that bridge that connects the two thalami together. And then underneath the thalamus, where it has all of these little guys here, that is the hypothalamus. It's a cluster and bundle of little neurons that are all kind of like contributing to your bodily physiological responses. Okay. Yeah, the red star tells you where it is. It's kind of a weird spot. Do I have it on this one? No. Okay, so pituitary gland. Remember I said the hypothalamus controls the entire endocrine system, meaning your hormones. We have 2 hormones that are actually in the central nervous system. So one is the pituitary. You can see that down here. It says pituitary gland. It kind of dangles underneath the hypothalamus. So we have thalamus underneath that, hypothalamus underneath the hypothalamus, the pituitary gland. It is considered the master gland. So it is the one that actually sends out signals to control all of your other glands, whether it's thyroid gland or adrenal gland to release adrenaline, or ovaries and testes, any of your other glands, it is the one that's controlling that. So hypothalamus, interprets what the brain thinks. I need to run away now. But then it tells the pituitary to actually produce the hormone that goes and activates adrenaline release. And we actually call that the HPA axis. Some of you might have encountered that before. The hypothalamic pituitary adrenal axis. So when we talk about stress, we'll get there. So don't worry about that yet. But know that the pituitary is the master gland. It controls all the other glands. One gland to rule them all. Okay. It's a Lord of the Rings reference. Okay. It's always more embarrassing when I do this in front of the class because I just get a whole bunch of blank stares back and I realize I'm a little too nerdy for these references. Okay, the second gland that is in the brain is the pineal gland. So here it's showing really nicely where the pituitary is. This weird bulby thing dangling down. This isn't the best representation because your eyes, I guess it's not the worst. Pituitary, the most kind of ventral portion. If you just flipped the brain and looked on the bottom side, you would see the pituitary. But the pineal gland is kind of more in the back. It's almost dead center in the brain, to the point that Aristotle actually believed that this was what controlled the brain was the pineal gland, and he thought it was like a joystick, like you like move the pineal gland around and then that moves fluids around in the brain. Very antiquated view, but tells you kind of where it is, almost smack in the middle, right? Thalamus is very much in the middle. Kind of behind thalamus is where we're going to find, or I should say posterior, is where we're going to find the pineal gland. And the main thing to know about the pineal gland, it produces melatonin. Melatonin makes you sleepy. So melatonin is responsible for us resetting our circadian rhythm every single day. When you wake up in the morning, your melatonin drops dramatically because you open your eyes and sunlight comes in, or your phone light comes in your eyes. Light entering your eyes reduces melatonin production. So the pineal gland stops producing melatonin. But throughout the day, especially in the afternoon, if you haven't had any coffee, that melatonin starts rising until by the end of the day, it's fairly high. On top of it, whenever the sun goes down, if you remain in darkness, melatonin goes even higher. So the light input to your eyeballs is directly related to how much melatonin is being produced. There's other factors involved, whether you're going to have more or less melatonin, but basically light to the eye and melatonin production are one of the main chronicity setters for your circadian rhythm, meaning it sets your circadian rhythm to be at our 24-hour cycle. I think it's more appropriately like a 23.5-hour cycle, but I'm not going to be picky here. So you can also take melatonin over the counter. Some people do this as a sleep aid. Sleep aids should only ever be used short term. I feel like I have an ethical duty to say that. Or if you are flying to the other side of the world, right, and you need to reset your circadian rhythm because your brain thinks it's daytime, but really it's nighttime. So some people, to combat jet lag, will take over-the-counter melatonin. Okay, so those are the two glands. Hypothalamus controls both of these. You can see that here. Hypothalamus controls the pituitary gland and the pineal gland. Pineal only does melatonin. Pituitary does all of the other hormone control. Okay, so those are officially all of the forebrain structures now. What time is it? 46 minutes recording? Okay, I think I can get through the last structures, and then I think I'll leave it there. I was going to do cranial nerves, but maybe I'll record that another time. We will talk in person about it, but just for now, I'm going to wrap up the midbrain and then the hindbrain. Okay. Again, don't worry about this terminology, tectum and tegmentum. I'll tell you the 3 areas that you need to know. Okay, midbrain, very, very small region. 2 of these structures are inside of the midbrain, 2 are on the outside. So these first, the ones that are on the outside are the colliculi. So if you look here at the brain over on the right, where it's green, on the very back side here, there are 2 little bumps, and those are legitimate bumps. So I'll have another slide that I show where you can really see that. Again, there's 2 each, 2 on the top and 2 on the bottom. The ones on the top, the superior colliculi, are for visual reflexes, right? So if all of a sudden there's like a flash next to you, you'd be able to very quickly orient and defensively orient to where that's coming from. Or if someone drops a book behind you, right, you'd be able to very quickly orient and figure out where that noise came from. Think of it as a defensive response, right? You very quickly want to know, reflexively react to potentially threatening things happening in the environment. So the superior colliculi, it's like the most adorable brain word ever, a colliculi. The superior colliculi is for visual reflexes, and the inferior colliculi, meaning underneath, are for auditory reflexes. So you can actually have somebody, we did that where we talked about brain dysfunction, right? For example, if you had a tumor or some other congenital defect in your visual cortex and you can't see, but your colliculi are intact, you would be blind, but you could still actually respond to very quick, rapid visual things happening around you. And, as far as awareness goes, you might not be able to talk about it very well, but a deep part of your brain registered that, and you'd be able to respond to it. Okay. The second one, we encountered this when we did our neurotransmitters, substantia nigra in the midbrain. So this is our main production site for dopamine. There's another one that we'll encounter again later, the ventral tegmental area. So 2 main areas for dopamine. Substantia nigra makes dopamine and projects it to basal ganglia so that we can initiate and control motor movements. This third one we haven't encountered yet. This is the periaqueductal gray. So aqueduct, if you remember our ventricles, we have the lateral ventricles. They flow into the third ventricle. And then the third, this is cerebral spinal fluid. The third ventricle via the aqueduct, the cerebral aqueduct, flows into the fourth being all the way down here, kind of in front of cerebellum. So the periaqueductal gray is a tissue where the aqueduct is. It's gray matter, so neurons that are peri around the aqueduct. So if we do a slice through the midbrain, this right here is the cerebral aqueduct. Let me make my cursor really big. This right here is the cerebral aqueduct, and this tissue around it is the periaqueductal gray. Very important for pain perception, right? Whether it's from your body going up the spinal cord or something on your face, but it is one of the only areas of the brain where we process pain. The second thing that I mentioned here are species typical behaviors. This could be a lot of different things. Probably the easiest to think of are grooming behaviors, right? Humans, we like to bite our nails or play with our hair or maybe scratch our scalp, or some people pick hair, which is called trichotillomania. What else? As far as humans go, I don't know how many human typical grooming behaviors we have, but think of any other animal, right? If you have cats or dogs at home, you've probably watched them groom before. My cat loves to do his back paw nails, and he's just picks at them, or when they're grooming their fur. These are all species typical behaviors, right? And there's not just grooming, there's other ones too. But I'm just kind of hand waving right now over them. But periaqueductal gray is really important for those kinds of things too. Because there's a really just long list when it comes to species typical behaviors. Okay, that was midbrain. That was it for midbrain. The colliculi, the superior and inferior, the substantia nigra for dopamine and initiating and controlling movements, and then periaqueductal gray for pain and for very specific behaviors related to the animal. Okay. All right, hindbrain, we're almost there. These are 3 areas of the hindbrain: the cerebellum, the pons, and the medulla. You don't need to say the oblongata. I don't think it's actually a proper term, but for some reason textbooks have it all the time. The medulla. So, or you can say the medulla, and it sounds like you're casting a spell or something. Okay, the cerebellum, this really weird looking guy, super wrinkly, does not look like the rest of the brain structures, is a very mysterious area. So cerebellum translates to little brain. Cerebrum would be the main brain, the forebrain. Cerebellum would mean little brain. It also has 2 hemispheres, just like the main portion of the brain. Used to be that we thought that it really just does kind of sensory motor tuning, right? Because it's so close to the spinal cord. It probably just takes in information and helps the brain process that information, kind of like an in-between step before it gets up to the brain. But it does so much more than that. So not just, it does do sensory motor integration, but it goes way beyond that. So any kind of fine motor movement that you have to do, whether you're an athlete and you have to be very careful where you put your foot and how you do that backflip, or musicians, knowing exactly, I used to play the violin, knowing exactly where to put your fingers, right? That comes with learning, but eventually, almost on the millimeter precision, you know where to put your fingers. Or an artist, any kind of fine motor movements, cerebellum is calculating these fine motor movements. So not just in time, but in space, right? Knowing for a baseball player to know either with a bat or with throwing exactly when to release the ball, that is like an incredible amount of tuning that happens. And that's not actually done by motor cortex. It's done by the cerebellum. And then it sends that information to motor cortex to make sure at exactly the right time and exactly the right spatial configuration you release the ball or you jump off the trampoline to do your backflip or whatever it is, right? So that's what time and space calculations means. Very, very fine tuning for motor actions. It does a lot more sophisticated things than that, actually. It also does planning and remembering, learning, experiencing emotions, language, social function. I mean, the list goes on. The more time passes, we developed fMRI technology in the 90s, and since then we've discovered more and more things that cerebellum does. It's involved in way more than we ever thought. So it really gives credence to the term cerebellum being little brain. It's almost its own thing that is doing a lot of heavy lifting for the brain. It's not just a little, additional auxiliary brain bumped on the back of your head. Okay. The other two areas, the deeper you go into the brain, the more it's important for basic survival. So pons, its main thing is sleep and arousal, right? So if you're asleep or if you're awake or if you're in a coma, what kind of coma, a vegetative state, half brain dead, whichever one, pons is determining that level of arousal, that level of wakefulness. Also does a lot of left and right body coordination because information's coming up from the spinal cord, and then it tries to coordinate and figure out which brain areas it's going to go to. That's all the pons does. The pons is over here, kind of what I think of as the belly of the brainstem. This up here is thalamus, pons, and then the last one is medulla. So medulla, these are clusters of nuclei that do a lot of very basic functioning, helping you breathe, helping your heart beat the way that it should, coordinating your breath and your heartbeat. So extremely vital for cardiovascular and respiratory centers and other areas, helping link up your internal organs there. We encountered the area postrema, your vomiting center. That's in the medulla. So again, it's not just for breathing and cardiovascular. It's heart rate breathing, but also swallowing, vomiting, urinating, gagging, and orgasming. So think the very, very basic survival functions, right? When we talked about hypothalamus and the 4 Fs, these are kind of a more holistic, how am I going to deal with the situation? Am I going to fight this person? Or am I going to mate with them? Or am I going to run away? Or am I going to eat this cake, right? But down in the medulla, it's doing way more basic, think almost like reflexes, right? We need to make sure we swallow that food. We need to make sure that we don't gag and vomit it back up. We need to be able to properly, breathe and heart rate and coordinate all that. If we're going to be running away from a predator, then we need to help accelerate our heart rate and make sure that that's okay. When it comes to the 4 Fs, right? Mating, we need to, if we're going to procreate, we need to actually properly orgasm, all of these different things. So it's the even more basic survival functions. And on this last point here, we encountered this with the neurotransmitters. Midbrain, pons, and medulla, all three of these areas are major neurotransmitter production sites. So we encountered it with dopamine, the substantia nigra in the ventral tegmental area. We encountered it with serotonin, which is the Raphe nuclei. And the third one was basal forebrain, which is not actually properly brainstem, but there are other neurotransmitters that we didn't talk about in detail that are made in these big brainstem regions. So this right here represents what we refer to as the brainstem. Up here, the thalamus, the superior, I told you it was bumps on the brain. So midbrain, superior colliculi, inferior colliculi, and then inside of here, we're going to have substantia nigra and periaqueductal gray. And then this right here, pons cerebellum, would kind of be here, like slapped on the back of this whole thing, and then medulla. And these yellow-orange things coming off, these are your cranial nerves. So we're going to talk about those properly another time, not right now, because this is a 1-hour recording at this point. And I'm getting tired of talking. But that was the main neuroanatomy, because then we have spinal cord. I'll talk about that in person, but those are the main areas that I want you to know for the central nervous system. Lots of areas. We are working our way through it, but this way, when in doubt, if you have a question about one of them, hopefully you can go back and view that. Okay, guys, I'm going to stop it here because, like I said, it's 1 hour, which is roughly what our class time would be anyways. So hopefully that helps. Hopefully you can go back and review this so you can get a better idea of all the neuroanatomical regions and their functions. I mentioned this in my announcement that I sent out today. Be sure to focus on the functions. It's good to know where they are. Is this part of the midbrain, the forebrain? Where is it roughly situated? But above all else, know the function. That is the most important thing, because as we move on in the class, the function is going to be the valuable piece of information that we need to know about. Why is the amygdala important? Because it does this and this, right? It's good to know it's in the temporal lobe. It's good to know it's kind of like in the front. It's important to know it's the limbic system or part of the limbic system. But again, you really want to make sure you know what it actually does. Okay. All right, I hope that helps, guys. I'll be uploading this, and I will see you in class next time. Okay, bye.